So we can see one of our coenzymes here. So remember, the coenzymes are really useful for facilitating these redox reactions. So what makes the coenzymes useful in facilitating redox reactions? Well, it's because they can accept hydrogen and also donate hydrogen, depending on whether they're in the electron rich or the electron poor form. So essentially what's going to happen here is we're going to have the oxidation of the alcohol functional group, which we can see in ethanol at the top here. So we're going to have the loss of hydrogen from that alcohol functional group. Where is that hydrogen going to go? Well, it's going to go onto our coenzyme here. And the result of this is that the carbon will now need to form an additional bond. So not only is the hydrogen within OH lost, but one of these other hydrogens is also going to be lost. So two hydrogens come off ethanol and are going to be added on to the NADH. And this process is actually facilitated by the alcohol dehydrogenase enzyme, which is an enzyme that facilitates this reaction, and that is present in the liver. So remember, that's why we have to move the alcohol to the liver, specifically the ethanol. It's to actually have this enzyme ready to facilitate this process. And at the conclusion of the reaction, we've now got our new products. So we can see here that we've got a new aldehyde group formed. So in the absence of these two hydrogens, what you would actually have is this arrangement, because the extra bond to the hydrogen down the bottom here has been lost by the carbon in the center here. So what does this mean for the carbon? Well, it means that it needs to form an extra covalent bond to be stable. What about this oxygen? Does it have enough bonds? No, it doesn't. It needs two covalent bonds as well. So the carbon here and the oxygen here both need an extra covalent bond. So what's the solution to this? The formation of a second bond between those two. So in doing this, we've actually created a carbonyl group, and the carbonyl group is actually attached to a hydrogen here as well, which makes the aldehyde functional group. So this shows you how loss of the hydrogen atoms from ethanol actually creates the aldehyde functional group within the acetaldehyde compound. You can also see the change that's happened to our NAD+, which has gained the hydrogens. It's now in the hydrogen-rich form, which is also electron-rich, and we see that as NADH. So it's accepted those hydrogens from the ethanol. So you can see summarized here in red that we've got an oxidation of ethanol as it's losing hydrogen, and that must mean the other component, NAD+, is being reduced. So we've got the NAD+ accepting the hydrogen there to become NADH. And we've got the creation of that new aldehyde group there shown in green. So essentially, by undergoing this redox reaction, we've done a functional group switch from the alcohol functional group to the aldehyde functional group. So this is a functional group switch facilitated by oxidation. So you can say alcohol can be oxidized to aldehyde group. Alright, so now we have an animation to show this a little bit more interactively, how things are moving around. So in this case, the OH group is being shown as oxygen bonded to hydrogen, which is the more technically right way of showing it. Normally we just summarize it to OH for simplicity. But I've drawn it in this way because that bond will actually be broken in the course of the reaction. So starting off with ethanol and our electron poor coenzyme, we then have a breakage of some of these bonds here. So it's going to be this bond here and this bond here as part of the oxidation of ethanol. We're going to lose those bonds, and those hydrogens are going to go over and associate with the coenzyme now. So as a result of this, we now have a carbon that needs an extra bond as well as an oxygen. And we have NAD plus associating with extra hydrogens. So that's going to allow each of these compounds to change. So we have our coenzyme now in the form of NADH in the electron-rich form, and we've got that extra covalent bond forming between the carbon and oxygen to satisfy the bonding arrangements of those atoms. So essentially we have the ethanol being oxidized, losing those hydrogens, meaning an extra covalent bond is needed between the carbon and oxygen. So this is what is required to convert the ethanol to the acetaldehyde compound. Alright, so the next thing we're going to have a look at here is the second redox reaction of the alcohol breakdown. So because it's the second reaction in the process, the product of the previous reaction which was the acetaldehyde, is now going to be a reactant in the next process. So it's a continuous process. You're going from step number 1. So the first reaction you can think of as converting A to B, and then the next step is converting B to C. So because A is being converted to B, initially B is the product of the first reaction, but in the next reaction B actually becomes the reactant to get converted into C. So you can see in a series of chemical reactions, what was previously the product is now going to be the reactant in the next reaction. And this is something we see a lot of in metabolic pathways, because a metabolic pathway isn't just a single chemical reaction, it's actually a series of chemical reactions. So whatever was the product of the previous step actually becomes the reactant in the next step. That's why they're called metabolic pathways, because it's a series of chemical reactions that work together to create a final end product that is needed for some purpose. So the final end product of this process that we're going to be creating is acetate. So you can't go directly from ethanol to acetate. We need to go through this intermediate, which was the acetaldehyde, and that's why we needed to do two separate chemical reactions. So just like the first reaction in this process, it's another redox reaction where the compound, in this case acetaldehyde, is going to be oxidized. So because it's being oxidized, the functional group is actually going to be altered again. So in this case, what's going to happen is the aldehyde will actually lose its hydrogen that's remaining here within that functional group. So remember, we've got the functional group at the end here, and it's going to lose a hydrogen, which is going to be given onto this coenzyme here. So this reaction is actually facilitated by another enzyme, acetaldehyde dehydrogenase. So again, this enzyme is only present in the liver, and that's where we need to have these reactions occurring, which is why the ethanol was originally taken to the liver in the first place. So let's have a look at the consequence of acetaldehyde losing this hydrogen atom by looking at what is in the product. Well, now it doesn't have hydrogen attached, it actually has OH attached instead. So because the hydrogen was lost, there's actually now space for the carbonyl group within the acetaldehyde compound to attach to something else. So we've got this spare bonding position. And it turns out what it bonds to is actually OH. So where do you think this OH might come from? Well, it actually comes from part of a water molecule. So the OH will actually split apart from this hydrogen and come in and become part of the functional group here. So in this way, the oxidation and loss of that hydrogen from the aldehyde functional group is actually allowing us to create a new functional group by the carbon accepting the OH group, and therefore creating a carboxylic acid functional group, which we can see here. You can also see that the coenzyme goes from the electron poor form to the electron rich form by accepting hydrogens, not only from the aldehyde but also from water. So we've got the acetaldehyde losing hydrogen, which is the main one we're focusing on. It's being oxidized to the acetate compound, and the component that's gaining hydrogen is our coenzyme being reduced to NADH. So here we can see the movement of these different parts of the water molecule. So the part in the green circle here is actually what's incorporated into the compound and forms part of the carboxylic acid functional group, whereas the other hydrogen actually gets donated to NAD+. So that goes there. Alright, so let's have a look at a short animation to see how all this comes together. So we're starting off with the acetaldehyde and also our electron poor coenzyme. So whenever you see one of these coenzymes like this, you automatically get a hint that it's actually a redox reaction, because they're the types of chemical reactions that these coenzymes facilitate. So if you ever see an NAD plus or an FAD and you're wondering what type of chemical reaction it is, you can remember that they're always present in redox reactions. So there's going to be oxidation and reduction taking place if you see one of these. So the first thing that happens is we're going to get a breakage of some of these bonds. So we're going to get a breakage of the bond with an energy input between the carbon and hydrogen, and also this bond here between the carbon and oxygen. And that allows those hydrogens to come off the compounds they were associated with and associate instead with the coenzyme. Because remember, that's what NAD plus does. It accepts the hydrogen atoms to be reduced in the redox reaction. And because our acetaldehyde has now lost the hydrogen, we can say it's been oxidized. So there's two components here that need to form an extra bond. This oxygen needs to form an extra bond within OH, and the carbon also needs to form an extra bond, which is part of this carbonyl. There's missing a bond here. So what could these two components do to facilitate a arrangement where both components are stable? Well, we can have the OH group coming close enough to the carbonyl group to form a new chemical bond, as shown here as the purple, and that will now mean that we've got a new functional group that's being created. So which functional group has been created by the formation of this new bond here in purple? We've got a carbonyl group and an OH group together. So which functional group is that? That's the carboxylic acid functional group. So essentially what we've done here, starting with the acetaldehyde compound that has the aldehyde functional group, we've converted that into a carboxylic acid functional group. And this has been facilitated via an oxidation. So this is the second reaction in the alcohol breakdown reactions, and that's oxidizing an aldehyde to a carboxylic acid. So we can also add in what we saw in the first step, which was starting off with an alcohol functional group that actually was being converted to an aldehyde, again via an oxidation as part of a redox reaction. So you can see this summary here of what's actually happened in these two alcohol breakdown reactions. So initially, the alcohol functional group is broken down or oxidized to an aldehyde group. And then the second functional group change is conversion of aldehyde to carboxylic acid, again facilitated by a redox reaction, in particular an oxidation. So why was it useful to actually go through these redox reactions to create our new carboxylic acid compound, which is acetate here? Well, remember I told you that alcohol was toxic and acetaldehyde was even more toxic. So by going through these two redox reactions, we've actually been able to create a compound that's now not toxic at all. Acetate can actually be used by the body, even turned into energy. So we've been able to take something that was actually toxic and potentially causing harm to the body in the form of ethanol and convert it into something very useful called acetate, which can actually be converted into acetyl-CoA and used to make cellular energy in the form of ATP. So now we're going to have a look at a few of the more technical parts of alcohol and why it's actually metabolized differently by different individuals. in general, it follows the same pattern that I just showed you in terms of using these redox reactions, but depending on the individual, they may not be able to perform these reactions as quickly as some others, and we're going to have a look at why this is. So as we know, alcohol is toxic to the body, and the amount of alcohol in the blood, or the concentration, is actually going to have a direct effect on how inebriated someone feels when they're drinking. So this is what they test when you do a breathalyzer, your blood alcohol concentration. So the more alcohol in your blood, the higher you're going to read on that breathalyzer test. So what the body is doing to counteract this alcohol being in the blood is it's going to be taking it to the liver where it can be processed. So essentially, as the liver processes and breaks down the alcohol, then the individual is going to feel less and less inebriated, or less and less drunk. And why is it taken to the liver? Well, it's because the liver contains these special enzymes that I showed you in the previous slides that are actually needed to facilitate those alcohol breakdown reactions. So in general, there's a standard rule that most individuals will break down around 1 standard drink per hour after they've stopped drinking. So this will vary a bit depending on the individual, depending on things like whether someone's eaten or not, your height, your weight, your ethnicity. So there's a lot that comes into it. So although you can use this as a rule, it's not going to be that reliable, but it's useful to have some idea of how quickly alcohol is being broken down. And as we know, we've got these redox reactions in the liver that are able to convert specifically ethanol into acetate. So remember, ethanol is the alcohol-containing compound. And once we've got that acetate, which is our end product of the alcohol breakdown reactions, that acetate can then be converted into acetyl-CoA, which can be used in a variety of metabolic processes which are useful. So we're going to focus a little bit more on these enzymes now, and the key to why we're doing this is because these enzymes are absolutely essential to facilitating alcohol breakdown. Without these enzymes, the reactions don't happen. So the function of enzymes in the body is essentially to make sure chemical reactions happen often enough to keep us alive. There's an enzyme that facilitates each different chemical reaction that we rely on in our body. So enzymes are actually proteins. So without these enzymes, essentially the chemical reactions that go on in our body, millions of them every second, wouldn't be happening. So essentially without enzymes we wouldn't be alive. So something to think about. So keep that in mind as we talk about how the presence of these enzymes actually impacts how well someone is going to be able to break down alcohol. So initially in the alcohol breakdown reactions, we have the toxic ethanol compound being converted into an even more toxic acetaldehyde compound via oxidation. And we can see that that reaction requires an enzyme called alcohol dehydrogenase. We can see that down here. So without the alcohol dehydrogenase enzyme, this process won't occur. It will occur at such a low level that you might as well not even consider it. So we need that enzyme for the process to be facilitated. And now once we've got that acetaldehyde, we then oxidize that further in a second redox reaction where the acetaldehyde is converted into the non-toxic acetate. And this requires a second enzyme called acetaldehyde dehydrogenase. So each chemical reaction requires a specific enzyme that's unique to that chemical reaction. So without either of these enzymes, the alcohol breakdown process would not be working properly. So essentially, through the reactions that are facilitated by these enzymes, we're able to break down the toxic ethanol into something that's non-toxic, and the body can use it. So everything going well, the reactions take place, the alcohol is broken down, and everything is great, so there's no problems. But what would happen if you didn't have as much of one of these enzymes as you needed to break down the alcohol? Or maybe one of the enzymes is there in quite a large amount, but it just doesn't work very well. Well, then we run into problems. And we can have a look at some particular individuals who are going to run into this problem more than others. But before we get there, I just want to make you aware that it's actually acetaldehyde, which is the compound that's actually responsible for most of the what people would call the undesirable effects of drinking alcohol. So that's the headache, the nausea and the vomiting, the dehydration. Most of these can be tracked back to acetaldehyde in particular. So although ethanol can contribute to some of these, the majority of the effect is coming from the acetaldehyde. So if this is building up in the blood, then you're going to be getting these symptoms. Quite common the morning after a drinking session for people to be having some of these symptoms. And that's because it's taking quite a while for the body to break down the acetaldehyde within the body. And obviously the more you drink, the more ethanol you had, and therefore the more acetaldehyde you'll have in the blood as well, because in the process of breaking down the ethanol, it has to get converted into the acetaldehyde first before it can get converted into acetate. So there's a limit to how much of these compounds can actually be processed by the chemical reactions, because there's only a certain number of enzymes that can facilitate these reactions. So we can't process all the ethanol at once, just like we can't process all the acetaldehyde at once. So essentially what this means is if you drink too much, then some of these compounds will still be building up in your blood even the morning after you've finished drinking and feeling some of these hangover symptoms. So an interesting question to consider now. Why do people of Asian ethnicity often get a red flush when drinking alcohol? And we can actually trace this back to these alcohol breakdown reactions. In particular, this enzyme, acetaldehyde dehydrogenase, because these individuals actually have a less efficient version of this enzyme. They've got enough of the enzyme in general, but it just doesn't work very well. So it's not as good at facilitating the chemical reaction from acetaldehyde to acetate than, for example, the version of the enzyme that is made by a gene in a Caucasian individual, for example. So you can say that Asians often have a gene variant that creates the acetaldehyde enzyme that is less efficient. So genes actually code for specific proteins. So in this case, there's going to be a gene that codes for the acetaldehyde enzyme, and if an individual has a particular variant of this that is a low efficiency version of that enzyme, it's not going to work as well. So essentially what this does is it slows down the conversion of acetaldehyde to acetate, so that last chemical reaction of the alcohol breakdown process occurs more slowly than it would, for example, in a Caucasian. So can you think of what one of the consequences of this might be? Well, essentially it means they're going to be getting more of these problems with acetaldehyde build up more quickly. So they're more prone to headaches and dehydration, also nausea and vomiting. So because the acetaldehyde will build up more quickly in these individuals, they can't drink as much before they're going to feel these symptoms. So where does the reddening come into this? Well, the other thing acetaldehyde does is it will actually promote dilation of the blood vessels. So this means that more blood can actually flow through the blood vessels because they're wider. And this causes reddening of the skin. This is often pronounced around the face, so this has got a bit of a nickname of being the Asian flush. Although some Caucasians and other people can get it as well, it's most common in people of Asian ethnicity. And it all comes back to the fact that these individuals will have an enzyme that essentially is less efficient and therefore not as good at facilitating the chemical reaction. So one of the other things that can happen due to acetaldehyde is an increase in the heartbeat. So the heartbeat can start beating quite rapidly, and that's another hint for these individuals that they should probably stop drinking and try and let the body do its job of breaking down the ethanol and the acetaldehyde and not introducing any more into the system. Interestingly, in general, women also have less acetaldehyde enzyme than men, which basically means that women break down alcohol more slowly than men. The version of the enzymes that women have are still going to work as well, there just aren't as many of them. So the conversion from ethanol to acetaldehyde and then from acetaldehyde to acetate takes longer. So among other things, this is why a woman who drinks, for example, 4 standard drinks will take longer to go back to a blood alcohol level of 0 than a man would, because less of these enzymes that are needed to facilitate the chemical reactions are actually present. So we have an interesting participation plus question to consider now about this content I've shown you with alcohol breakdown and how these redox reactions facilitate functional group changes, and also thinking about why some individuals aren't able to break down alcohol as efficiently as others. So I encourage you to have a go at this question, share your answers with other people in your cohort, posting on the Loop or using other means that your online tutor has told you that are available to try and share your answers and get an understanding of how different people have approached this question. These questions are quite interpretive, so it's common that people will come at it from a lot of different angles. And it's a really good opportunity to get you thinking about what you've just seen and work out whether you have understood what we've been talking about. So we've also got an opportunity now to do some of the conceptual multiple choice questions. So please have a go at 3 to 6 in your review quiz on the LMS, and that'll help cement the content that we've had a look at so far in this session. Alright, so to summarize this section, we had a look at redox reactions, and the common thing about redox reaction is that one reactant is always going to be oxidized and the other will always be reduced. And this is happening simultaneously. You can't have one without the other. So whenever you have one component being oxidized, you know something else has to be reduced. And we saw that there was this handy way of remembering what's being oxidized and what's being reduced in terms of what's happening. So oil rig tells us that oxidation is loss of hydrogen atoms, whereas reduction is gain of hydrogen atoms. So to work out which reactant is being oxidized, look at the reactant and look at it on the product side, and then work out whether it's gained or lost hydrogen atoms. So the one that's lost hydrogen atoms will be the one that's been oxidized, whereas the reactant that's gained hydrogen atoms to become its product will be the one that's been reduced. Then we had quite an in-depth look in the way that the alcohol was being broken down in the liver. As we saw, this relied on 2 redox reactions. So the first of these was where ethanol was being oxidized to acetaldehyde, and this corresponded to a functional group change from an alcohol to an aldehyde functional group. The second redox reaction was conversion of acetaldehyde to acetate, again an oxidation, where the aldehyde functional group was being oxidized to the carboxylic acid functional group. We also saw the key role of the coenzyme NAD+ in removing hydrogens from the ethanol and acetaldehyde compounds to facilitate oxidation in these redox reactions. We also talked a bit about the final product, which was acetate, the fact that it could actually be used by the body and it could be converted to acetyl-CoA and then be converted into cellular energy through different metabolic pathways. And the last part of what we looked at was the role of specific enzymes in these alcohol breakdown reactions. I showed you that these enzymes were absolutely essential to the reactions taking place, and if an individual had a less efficient version of this enzyme, it impeded their ability to break down alcohol, and more of the symptoms like nausea and headache would come on more quickly for these individuals. And it was individuals of Asian ethnicity that are most likely to have the genetic variant of the enzyme that means that they're going to be unable to break down alcohol as quickly as a Caucasian would.